In my previous video on enzyme kinetics, I've introduced several different types of enzyme inhibitors. Many drugs are enzyme inhibitors that target an enzyme to correct a pathological condition. In this video, I'll give examples of drugs that function as competitive inhibitors, suicide inhibitors, and transition state analogs. To recap, competitive inhibitors are substrate analogs that reversibly binds to the enzyme active site. Competitive inhibitors can function as antibiotics by inhibiting the synthesis of DNA. For example, sulfonamide is a competitive inhibitor of dihydroepiderase synthetase, preventing the addition of p-aminobenzoic acid or PABA to dihydroepiderate diphosphate, an early step of folic acid synthesis, which is critical for bacterial DNA production. Trimethylprim is a competitive inhibitor of dihydrofolate reductase, preventing the conversion of 7,8-dihydrofolate to tetrahydrofolate, the active form of folic acid. Trimethylprim and sulfonamides inhibit different steps of folic acid synthesis and have a synergistic effect when taken together. Competitive inhibitor of dihydrofolate reductase can also be used to treat cancer. Methotrexate and aminopterin are folate analogs, binding to the active sites of dihydrofolate reductase, preventing the synthesis of tetrahydrofolate. This results in the inhibition of DNA synthesis, which is used to treat rapidly dividing cells in cancer. Ibuprofen is a non-steroidal anti-inflammatory drug, or NSAID, that treats pain, fever, and inflammation. It is a competitive inhibitor of cyclooxygenase 1 and 2, also known as COX-1 and 2, preventing the conversion of arachidonic acid to prostaglandin H, which is a precursor to several pro-inflammatory prostaglandins. Allopurinol is a competitive inhibitor of xanthine oxidase that treats gout by blocking the conversion of hypoxanthine to xanthine, a step in purine nucleotide degradation. Allopurinol prevents the accumulation of uric acid in our body, thus lowering the chance of gout. Sildenafil, sold under the brand name Viagra, is a competitive inhibitor of phosphodiesterase 5, or PDE5. That treats erectile dysfunction by preventing the conversion of GMP to cyclic GMP. Normally, nitric oxide binds guanylate cyclase receptors, which results in increased level of cGMP, leading to vasodilation of smooth muscle. This signal transduction pathway is terminated by PDE5. Therefore, sildenafil prolongs vasodilation of the penis by inhibiting PDE5. Oseltamivir, sold under the brand name Tamiflu, is a competitive inhibitor of neuraminidase that treats influenza by preventing the cleavage of sialic acid, also known as N-acetyl-neuraminidase acid, which is found on glycoproteins on the surface of human cells that help new virion exit the cell. Therefore, oseltamivir essentially prevents new viral particles from being released. Acyclovir is a nucleoside analog that mimics guanosine and inhibits DNA polymerase of herpes simplex virus, thereby treating viral infection. Zidovudin, abbreviated as ZDV, also known as azithromycin or AZT, is a thymidine analog that inhibits reverse transcriptase, treating HIV/AIDS infection. Some drugs function as suicide inhibitors, also known as mechanism-based inhibitors, which irreversibly bind and modify enzyme active sites. Penicillin is an antibiotic that functions as a suicide inhibitor of DD transpeptidase, inhibiting the cross-linking of peptidoglycan, which is the final step of bacterial cell wall synthesis. Since human cells lack cell wall, penicillin specifically kills gram-positive bacteria. Aspirin, like ibuprofen, is a non-steroidal anti-inflammatory drug NSAID that functions as a suicide inhibitor of COX-1 and 2 through the acetylation of a serine residue in the active site. Fluorouracil, also known as 5-FU, is an anti-cancer drug that functions as a suicide inhibitor of thymidylate synthase through its active metabolites such as fluorodeoxyuridine monophosphate or FDUMP, blocking the synthesis of pyrimidine thymidylate or DTMP, which is a nucleotide required for DNA replication, thereby affecting rapidly replicating cancer cells. Vigabatrin is an anti-seizure drug that functions as a suicide inhibitor of GABA aminotransferase, abbreviated as GABA-AT, resulting in an increased level of GABA in the brain. The increase in the inhibitory neurotransmitter GABA puts a brake on the excitatory processes that can initiate seizure activity. Difluoromethylornithine, abbreviated as DMFO, is a suicide inhibitor designed to treat sleeping sickness caused by parasitic protist trypanosome. DMFO irreversibly binds to ornithine decarboxylase and provides an alternative electron sink, forming a covalent complex with the enzyme active site. Lastly, transition state analogs are the most powerful enzyme inhibitors because they fit into the active site stronger than the substrate itself. Anti-HIV drugs such as indinafir, nalfenafir, lopinavir, and saquinavir mimic the tetrahedral intermediate of HIV-1 protease, inhibiting the enzyme's ability to cleave polypeptides into active infectious proteins during HIV replication cycle.